Welcome to A Moldering in the Grave, what decaying dinosaur bones tell us about the age of the Earth. Millions and millions of years ago, dinosaurs roamed the Earth until about 66 million years ago, when dinosaurs had the ultimate bad day. When an asteroid struck planet Earth, bringing a reign which had lasted 180 million years to a dramatic end. Or that's how the story goes. So you see this story everywhere you turn. You see it in documentaries, in natural history museums. Here's the UK Natural History Museum. And they say how an asteroid ended the age of the dinosaurs 66 million years ago. That sounds familiar. Dinosaurs had the ultimate bad day. With a devastating asteroid impact, a reign that had lasted 180 million years was abruptly ended. This is so common, all the scientists believe it. You would have to be a little bit crazy to believe otherwise. So permit me for the next 50 minutes to be a little bit crazy as we explore a moldering in the grave, what decaying dinosaur bones tell us about the age of the Earth. So, a little bit on my background first. Um, my PhD is in molecular biology and genetics. I am not a paleontologist, but I would call myself a molecular paleontologist.
Medical.
Molecular biology is also known as tiny stuff that you can't see without a very expensive microscope. So I did my research in Alzheimer's disease in fruit flies. During my master's degree, during my PhD, I studied development, which I think is the single most incredible aspect of all creation. It's how a single cell goes into a full-grown organism, and all of that is programmed into that first cell. And I'm not going to talk about any of that tonight. My family and I live in sunny Arizona. I'm blessed with a whole quiver full. I have 8 children, number 9 on the
Hey,
way. And
wait.
here's what I thought having a large family would be like. Here's what it's actually like. It's a little more like Jurassic Park, with all those kids running around. We have a great time. We moved to Arizona. By the way, if you are in the Zoom, be sure to mute your mic. If you are viewing this online, mute your mic. We moved to Arizona 2 years ago, where I became the director of the Creation Research Society. What's the Creation Research Society? It's a team of creation scientists. It's the largest and the oldest network of creation scientists in the world. We have over 600 scientists that are members of the Creation Research Society. We do four things. We publish research. So we have a journal, a technical peer-reviewed journal that we publish every quarter. We present research. So kind of what I'm doing here tonight, presenting some of the research that we are doing, but also we have an annual conference. This year it's in Pensacola. So whenever you get tired of the cold weather up north, although it's going to be beautiful up there, I'm sure, in July, uh, we have our conference in Pensacola this year. And this is a technical conference. So you'll have creation scientists presenting kind of their latest research. We fund research. So if somebody thinks they have the next greatest idea in creation science, they can submit that idea and try to get a little bit of funding to get that off the ground. And we perform research. This is what I'm here to talk about tonight, is some of the research that we perform in our lab. Our lab is called the Van Andel Creation Research Center. We're based on the campus of Arizona Christian University in Phoenix, Arizona. Uh, I'm going to put this QR code up again at the end, but I encourage you, join the movement. Creation scientists are vastly outnumbered by our secular counterparts. We need people working together, and that's what the society is all about, is getting people working together. And especially if you are a scientist out there, then check out the Creation Research Society. If you are just someone who's interested in the technical side of creation research, then you should also check out the Creation Research Society. I'll say some more, or I'll put this QR code up one more time at the end. But I'm not here to talk about CRS. I'm here to talk about dinosaur bones. Now, you see this beautiful AI generated dinosaur skull in the background. That's definitely not real. Uh, what can you learn about a thing from its dead bones in the ground? What do these decaying bones tell us, if anything, about how old they are? Well, when I started into this project, I'm coming from a molecular biology background. I'm not a paleontologist, but I started studying these dinosaur bones, and I was surprised at, in the grand scheme of things, how recently we've learned about dinosaurs. And I say we, I'm talking about in the modern era, how recently we've learned about dinosaurs. In fact, here is the first dinosaur ever named. This is Megalosaurus. So British scientist William Buckland, he's digging around. He finds this jaw. It looks like it's from a lizard, but it looks very big. So he calls it the Mega, for big, Saurus, lizard, big lizard. And this was 200 years ago, 1824. Here's what it looks like. We know now approximately. That's the Megalosaurus. That was just 200 years ago when they named the first dinosaur. Meet the second dinosaur ever named. This is the Iguanodon. And similarly, they find here's the publication when they reported on this interesting creature. They say, notice on the Iguanodon, a newly discovered fossil reptile. So they found the teeth first, and similarly, they thought these teeth look like a reptile tooth. They look like an iguana tooth, and so they named it Iguanodon, not knowing what this incredible creature was. And this was 200 years ago. So for thousands of years, these dinosaurs were just buried in the ground, unknown to modern humans, modern as in the last couple thousand. And then they start learning an awful lot. So here's originally what the iguanodon was thought to look like. These large, sluggish quadrupeds. If you look very closely at this particular reconstruction, this iguanodon has a spike on its nose. Well, that makes sense. You find a spike along with the skeleton, probably goes on the nose. So they stick it on the nose. Well, here's a modern reconstruction of the iguanodon. Here's what we believe it looks like now. And you just see the spike on the nose? Nope, you don't, because it's gone. Instead, the spike is on the ground. It's like a spur, a thumb spike that is on the foot of an iguanodon. The term dinosaur wasn't coined until 1841. So it's really been within the last 200 years that we have learned an awful lot about these fascinating, incredible creatures. And it's interesting to me to think about all the hundreds of years of people walking around with these bones buried under the ground, unknown to our modern existence. How do we know all about dinosaurs now? Well, it has to do with actual real field paleontologists. This is Art Chadwick. He's a creationist and a phenomenal paleontologist. Had the opportunity to meet him at the Dinosaur Science Museum there at Southwest Adventist University in Keene, Texas. They have just rooms filled with all these bones that they've dug out of the ground from a ranch in Wyoming. So all of those white casts, that is when they're excavating the bone. They put this white plaster around it to stabilize and preserve the morphology of the bone. And then they bring those all back down to their dinosaur research lab and start to clean them up. So they just got rooms filled with these bones. And it's through the work of paleontologists like this that we know an awful lot about dinosaurs. Here, I am holding a tooth from this guy. I can confirm the T-Rex is real. So we have found all these bones, and you would be right to ask, how did these bones get there? How did they get into their current fossilized form? There's a whole field of science that studies this. And this is the first and one of the only $10 words I'm going to give you tonight. So the field of science that studies how you get from, let me put my pointer on, how you get from the dead critter over here, all the way to the fossilized, mineralized bones buried in the ground over here. The field that studies that process is called taphonomy. Tapho means burial. Onomy means the laws of. So taphonomy is the study of burial. In this image over here, I've got these green spheres. The green sphere is the living thing, it's the biosphere. So taphonomy is the transformation, it's the study of the transformation of the organic remains from the biosphere. That means when something was alive, to the lithosphere. That means when it's converted into a dead rock, which we know as fossils. Another way you could sum all this up is taphonomy is the study of a moldering in the grave. Now, I hope it's obvious that as soon as we start talking about the past and like how did these bones get here? How long have these bones been here? As soon as we start asking questions like that about the past, we are leaving the realm of empirical science. I'm going to be showing you some empirical science tonight, some research that we do in our lab. You can use empirical science to partially answer those questions about the past, but you can never fully answer a question about the past with empirical science. When you start asking about the past, you are entering the world of world views, the set of assumptions, the belief system that you have that you use to interpret the data. So let's just set the stage right here at the beginning. I don't want to sneak up on anyone. You guys don't know me. I don't know you. But I am from a creation research society. So I'm operating from a biblical worldview, a straightforward biblical worldview that believes that God actually knows how to communicate and is able to communicate how he created this world in 6 days about 6,000 years ago. So in the biblical worldview, we are going to compare this to the evolutionary worldview and use these two worldviews to interpret the dinosaur fossils. When you look at the fossil record, you do find a pattern. This is a pattern that's consistent across the globe. There are certain layers that contain certain types of fossils, marine fossils towards the bottom, dinosaurs are here in the middle. When the evolutionary worldview looks at this fossil record, they interpret it as a progression of time. So you have the simple, and those things gradually get more complex over time. When the a biblical creationist looks at this pattern in the fossil record, we interpret it as a progression of the flood. So you have the vast majority of fossils are marine fossils that will dwell on the bottom of the oceans, and they were the first victims of the flood. So they form the bottom of the layers here. And then as you move up, you get into more, you get into land animals, the less mobile land animals, and eventually at the top, you have the most mobile land animals like the birds, the humans, things like that. Ones that aren't captured by the flood waters and fossilized in the flood layers. So we interpret this as a progression of the flood. Now, these are two different perspectives on it. And depending on which perspective you have, if we look at the age of dinosaurs, and dinosaurs are consistently found in these kind of middle layers, which would be towards the end of the flood in the biblical perspective. So when you find these dinosaurs in these specific layers, and you ask the question, when did the age of dinosaurs end? If you have a biblical perspective that says the flood was about 4,500 years ago, so the age of dinosaurs ended roughly 4,400 years ago. That's when they mostly died out, and that's when these fossils were formed. If you look at the evolutionary timeline, they would say that the age of dinosaurs ended about 66 million years ago. This is the asteroid story. All right. Things that are different are not the same. These are two very different answers to the same question of when did the age of dinosaurs end? Now, if you are holding the worldview that says it's 66 million years ago, then it would have come as a surprise in the early 2000s when Mary Schweitzer, she's the first author on this particular paper and has been one of the primary scientists studying this in the conventional science paleontology. She started digging up and looking into these T-Rex bones, and she found soft tissue. So unmineralized, stretchy, fibrous tissue. That's what you can see on the left there. On that middle panel, E, you've got these vessel-like structures. And as scientists, we try to be careful in the language that we use. We're not saying they're blood vessels. She didn't say they're blood vessels, but that's probably what they are. Well, they're just called vessel-like structures. You even have things that look like red blood cells. These are not intact red blood cells like you would find in a modern organism, but they bear some similarities to a lysed, a broken red blood cells. So they're finding these tissues in dinosaur bones, which are reported to be 66 million years old. This, of course, caught creationists' attention, and the Creation Research Society launched the iDino Project. In the iDino Project, this stands for Investigation of Dinosaur Intact Natural Osteotissue. And I'm not going to say that again. So, in the iDino Project, several scientists went up to the Hell Creek Formation in Montana. This is a place where those middle layers, where the dinosaurs are rich, that are rich in dinosaur bones, those middle layers are exposed at the surface, and you can readily find dinosaur bones in these layers. And so they went up to the Hell Creek Formation and started digging around for dinosaur bones, and they found this guy, which was a beautiful specimen. This is 118 centimeters long, Triceratops browhorn. So I've got my little Triceratops guy right here. That's this. So the Triceratops browhorn encased in sandstone. And if you look very closely, these tiny filaments are called filopodia. They're tiny extensions of the cell that anchor it to the matrix that surrounds it. All that to say, these are beautifully intact cells found within dinosaur bones. They went back to the Hell Creek Formation and also dug up this Thescelosaurus vertebra. And when they dissolved the hard material, this is what was left behind. Some of this soft, stretchy, fibrous material found inside of these vertebra. So here is another example of soft tissue. Here's a video showing another vessel-like structure, like those ones I showed you earlier. So it's initially this was a very controversial finding soft tissue in dinosaur bones. Not so much anymore. It's been for the most part accepted by all scientists, including secular scientists, and because of the evidence. So here's what I mean by evidence. There have been now over 100 papers published describing this soft tissue and molecules in dinosaur and other ancient fossils. So there's pliable tissue, like those ones I showed, morphologically distinct cells, like those bone cells. Collagen, that's a protein. Collagen fragments are frequently detected in these tissues, and other proteins, such as actin, myosin, etc. So all of this evidence building up over the last 2 decades has led scientists to say, yep, it's there. There's soft tissue there. Here's one of the most recent papers. This was in 2025. Evidence for endogenous collagen in Edmontosaurus fossil bone. Now, I want you to pay special attention to that word collagen because collagen is the main character in the rest of our story tonight. So collagen is a protein found in these bones. What is collagen more specifically? What is collagen? What do you think of when you hear the word collagen? Maybe you think of an anti-aging elixir cream that you can put on your face and prevent those wrinkles. It works in your 30s, in your 40s, in your 50s, and beyond. This is very popular nowadays to take collagen supplements and things like that. Maybe you think of pork rinds with 12 grams of collagen per serving. Now, where is all of this collagen coming from? Well, it turns out collagen is the most abundant protein in your body and the most abundant protein in a pig's body and the most abundant protein in a dinosaur's body. So collagen is the most abundant protein in vertebrates, body. In order to understand collagen, I'm going to build a little piece of collagen before your very eyes. So I need to stop sharing my screen for a moment. Okay, here I am. Here, so I'm going to build a little piece of collagen. I'm going to start with this guy right here. This is an amino acid. It has multiple colors on here. Those colors have meaning. It's a molecule, and these are the individual atoms. So this is hydrogen in white. We've got nitrogen in blue. We've got oxygen in red, and the black is carbon. Carbon forms the backbone of all major biological molecules, which is why we're called carbon-based life forms. All right, so this is a single amino acid. This happens to be the smallest of all the 20 amino acids that make up your proteins in your body. This is glycine. A single amino acid does not collagen make, but I'm going to add another one in here. Okay, so here comes amino acid number two, amino acid number one. They're going to connect together, and now I'm the beginnings of a chain of amino acids. This is alanine that I just added. Let me add one more. Here comes the next one. It's coming in here. I'm going to attach it right there. I just added a third one, and now I have three amino acids in a chain, and this is a small, tiny, tiny fragment of collagen. This is what collagen would look like. It's just a chain of amino acids, but it's a chain with special properties. So let me go back to the big Oh, wait, 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 wait. One more thing I want to show you. Okay, I'm going to have to hold this up close. Do you see that purple bond right there? That purple bond is what I want you to focus on for the rest of this talk. It is the bond that holds the amino acids together. And it's called a peptide bond or an amide bond. And we're going to be talking about those a lot. So that purple bond, that's really important. All right, screen is coming back. So here's what I was just building for you, a small piece of this. I built three of these amino acids. That's what collagen is. It's a chain of amino acids. And see those red dotted lines? Those are important because those are marking the peptide bonds that connect the amino acids. Now, collagen is a very, very long chain of amino acids. Here's the full length, and I'm going to be quizzing you on this at the end. So start writing. This is hundreds of letters long. And I'm going to zoom in on a portion of this chain, and I want you to see if you notice anything interesting about the pattern. I'm going to give you 10 seconds to quietly look. Okay. Did any of our brave souls online want to unmute and tell me something you see in this string of amino acids? Anybody notice a pattern? It looks like I don't have any takers. I'm guessing somebody in the audience knows, but I'm not gonna try to unmute anything there. All right, I'm gonna highlight something to make it even a little simpler. You notice those three letters, GAP, those are actually the three letters I built for you. That's the GAP. But did you notice that this is a common pattern? Not the A and the P, but the G. Every third letter is a G. That's not by accident. It's actually designed that way. G, glycine, is the smallest amino acid, and because it's every third letter, collagen is able to be braided into a very tight, highly stable structure. So what you're seeing here is actually three molecules of collagen. That region in the middle, right here, this is what I built for you earlier. And the rest of it is the full chain of collagen. It's beautiful. Now, it's not actually pink, green, and blue inside your body. But this is three chains and they're braided together into a very highly stable structure. And this is good because collagen holds your body together. When collagen starts breaking down, guess what you get? You get wrinkles. Right? So you can be grateful that collagen is such a stable molecule. But what does collagen have to do with bones? I have here a bone which I extracted from an alligator. I'll tell you more about that later. This is a bone from an alligator. This alligator is dead, in case you didn't know. And inside the bone, this bone is loaded with collagen, believe it or not. It's loaded with the hard stuff, the mineral, but it's also loaded with collagen. So, exactly what does collagen have to do with bone structure? If you were to look inside the bone, here's what you would find. It's made up of these two parts, the hard part, the mineral component, and then the organic part or the proteins, and that's the collagen. So the collagens are like these fibers that the mineral attaches to. You can think of it just like this. It's like rebar reinforced concrete. So you've got concrete, which has extremely strong compressive strength, and then you mix some rebar in there, and it gives it tensile strength. So this is the way the bone is designed. This was actually God's design before it was our design. Your bone is just like this, where the collagen is like the rebar, and the mineral is like the concrete. If you were to take a bone and split it up into its pieces, it's about 10% water, 60% bioapatite, that's the mineral component, and about 30% organic. So that pink slice is the organic slice, and that includes collagen. Collagen is actually the most abundant component in that organic section. So about 27% of the bone's weight is actually collagen. Which brings us to a most interesting question, and the question of the evening. How long can collagen lie moldering in the grave? Let me go back to our little bone right here. So I told you this bone has collagen in it. This bone is from an alligator that's now dead after the alligator died. What happened to the collagen? Well, the collagen starts to break down. And eventually, once enough of it breaks down, the whole bone will dissolve and disintegrate and crumble. And it'll be recycled back into nature. However, this bone, does it still have collagen in it now? Yes, it does. How much? How long does it last? Those are questions we can start to answer with empirical science. So let me show you how we do that in our lab. We're going to focus on those peptide bonds. We're going to measure how long it takes for those peptide bonds to break apart. We're going to use this machine called a Fourier transformed infrared spectroscopy or spectrometer. And you can think of this just as a very expensive flashlight. And it shines a special type of light called infrared light. So here is the molecular structure of collagen. I'm showing you two amino acids and see how they're connected by that blue bond. That's the purple bond that I was showing you earlier. So this is the peptide bond. It's also called the amide bond, but that's the bond that we are measuring. That's the one we care about. Well, it turns out that when you shine infrared light at a peptide bond, it's going to absorb some of that light and it's going to transmit some of that light. And it forms a very specific pattern based on what light it absorbed and what light it transmitted. And you can measure this pattern, and it looks like mountains and valleys. So here's an example. In this particular example, you are seeing on the y-axis, you're seeing absorbance. That's how much light it absorbed. And on the x-axis, you're seeing the wavelength of light, reported here as wave numbers. But what I want you to focus on is the green line. The green line is a sample of well-preserved bone. And when you look at the green line, you notice, ooh, right here, this very tall mountain peak, that is the peak that represents the peptide bond. It's amazing that all you have to do is shine a light at it and you can detect whether there are peptide bonds in your sample. Compare that with the red line. The red line is poorly preserved bone, and you follow the red line along here, and in this same region, it's a very low peak. That's all we're doing is we are measuring mountain peaks. Here's what it looks like in our lab. I'm going to show you 3 lines on this graph. And this is special bone. This is bone that was treated with heat, a little bit of temperature, which is going to speed up the decay process. It's going to speed up the breaking of those bonds. So here, the blue line represents a bone which has not been exposed to heat, and it has a nice tall mountain peak. So those mountain peaks, this is the one we're looking for right here. It has a nice tall mountain peak. The green line has been treated with heat for 7 days, and the mountain peak has gotten smaller. The purple line has been treated with heat for 14 days, and the mountain peak has gotten smaller. So that's what we're measuring. We're measuring the decrease in the height of this mountain, and that represents the number of peptide bonds. Notice this other mountain over here, I'm not going to say much about it other than it doesn't change. So it stays the same. And so we use this mountain, the phosphate peak, to normalize our amide 1 peaks. Here's ultimately what we are measuring in this process. We're measuring this event, the breaking of that bond. When that bond is broken, the mountain peak drops. All right, so that's the science that's going on behind this. Now let me walk you through the experiment. We have done this in 3 different organisms. Mammalian organism, cow, avian, ostrich, and reptilian, an alligator. So here we are dissecting alligator legs to extract the bones from within. And it was a very messy process. You might be wondering, how in the world do you get alligator legs? I wish I could say I hunted these alligators myself, but it was much easier than that. We just called up an alligator farm in Florida and said, send us the largest legs from your largest alligators that you got. And a couple days later, they arrived in the mail. So we've done all these experiments using leg bones from these various animals. Here's what the process looks like. We take those bones and we crush them up into small fragments. These are about 1 millimeter in diameter. We then incubate those with temperature. So we expose them to heat. And the reason we do this is because the collagen is extremely stable. It would take hundreds of years at room temperature for the collagen to break down. It is that stable. And we want the experiment to be done before we die, so we accelerate it with heat. We then grind it into a fine powder. We run that fine powder through the FTIR, so we shine the light on it, and it creates the mountains and valleys. And then from those, we generate these decay curves. We use those decay curves to create something called an Arrhenius plot, and voila, we're done. So these are the Arrhenius plots. I'm going to explain this in a little bit more detail in a moment. For now, I just want to show you a video of how we generate these Arrhenius plots. So you can kind of get a behind the scenes look into the Van Andel Creation Research Center. There will be some audio background, just music. I'm going to be narrating it live as we go. This is the art of measuring bone collagen decay rates. So first, we get the bones from the animals, and then we put them through this rock crusher. That gets them down to a slightly smaller size. We then put them through a hand mill. This gets them down even smaller. Then we put them through a grinder, and this gets them down even smaller. We don't want to get them all the way to a powder, but we want to get them to a manageable fragment size. Okay, so now we have a nice homogeneous mixture of these bone fragments. The only thing you need to do to cause collagen to decay is it needs a little bit of energy and a little bit of moisture, both of which are abundant in nature. If you have bacteria, it will decay even faster. But right now we're just looking at the most basic form of decay. So we took 1 gram of bone water, and we add it to each of these vials. In this example, we're showing you 5 different vials, but in a full experiment, you would have anywhere between 50 and 128. Our biggest experiment had 128 vials. It was a handful. By the way, this is Ellie. Ellie is an undergrad who just graduated, worked in our lab for 2 years, and the undergrads here at Arizona Christian University that have been invaluable in helping to perform this research. All right, now the sample's going into a water bath. That's just what it sounds like. It's a bath filled with warm water, and we do it at a specific temperature. Okay, we then periodically, while that's in the water bath, we pull out a few samples. We do this every 3 or 4 days, and then we leave the rest to keep incubating. We're testing at different time points how much collagen has decayed. This student here, her name is Faith. She also just graduated, so we lost 2 really great students this year, but we've got more on the way. She was an undergrad going into medical school. She is rinsing these into this very highly technical instrument known as a coffee filter. What we're doing is we now want to dry these bones. We have to dry them before we can run them through the FTIR machine. Drying them is the easy. So we are doing this with not just 3 samples, but dozens and dozens of samples. This is what it looks like. The drying them is the easy part. The hard part is grinding it. In order to measure a sample on the FTIR, it has to be a very fine powder. We sped this up because the video would have taken about 15 minutes, real time. It takes a long time to grind it into a fine powder. Now what you're seeing is Faith is taking that powder and she's running it through a sieve. So this is going to collect just the right size. We need powder which is between 23 microns and 60 microns in diameter. And micron is a millionth of a meter. There is that finished powder. This guy ready to go get measured on the FTIR. So that brings us to the final step. We take out these samples. This is Brian. He's one of he's a lab tech, jack of all trades, works on several different things in the lab. This is the FTIR. So that little circle is where the light's going to come out. And we're going to shoot a beam of infrared light through the powder. You actually, it looks very boring because you don't see the beam come through. It's just going through that pedestal. And we have to do this with every sample 3 times. We have to do biological replicates of each experiment 3 times. And we collect all this data and put it into the giant spreadsheets. These are the heights of all these different mountain peaks. So we measure the heights of the different mountain peaks. We do some fancy math and use that to generate some graphs. Now, you don't actually want to be doing this on paper and pencil. We use a programming language called R. This is going to take all that data, it's going to analyze it for us, and it's going to create those Arrhenius plots like those ones I showed you at the beginning. Okay, and that's that. I wanted to show you that so that you could see kind of behind the scenes. When you see an image like this that I have on the screen right now, this represents a year and a half of experiment and work to generate these results. Now let's talk about what they mean. I'm gonna break it down for you. All right, so here, let's start with the ostrich. Here's what the experiment would look like. On the y-axis over here, this is the collagen concentration. So you see it starts pretty high. On the x-axis, this is time in days. And so over time, the line goes down. All right, that's not that complicated. So this is one example of one experiment. And you can see three three dots. Those are three replicates for each of these experiments. But we don't do it at just one temperature, and this is the critical part. We actually do it at four different temperatures from 90 degrees to 75 degrees. Now, here's what I want you to notice. Notice here at 75, it's kind of a flat line, and then it gets steeper, steeper, steeper. So as the temperature increases, the breakdown of the collagen, the speed at which it breaks down also increases. This is common in biochemistry. That's very normal. But we don't do it for just ostrich. We do it for ostrich, cow, and alligator. And this is what the full data looks like. I'm going to zoom in now on the cow because this is not the finished plot. This is just that first step. So we have these data, we generate all these plots. The thing that we really care about in these plots is the slope, which I've put in this red box. That's the slope. We take those slopes and we're going to plot those slopes against the temperature. That slope is the decay rate. So here are the decay rates at 85 degrees for cow. Here are the decay rates at 80 degrees for cow. And do you see what's happening? So this plot is showing us a relationship between the decay rate, which is very high at 90 degrees, and the temperature. So as temperature decreases, the decay rate decreases. And you can see where we're going with this. Now we can project what the decay rate would be at lower temperatures like room temperature, where these things fossilize. Okay, so we did all that work to get this slope right here. We're going to use that to go into this fancy formula. We compute using the ideal gas constant, something called the activation energy. This is the amount of energy that is required to break that bond, that purple bond. And the activation energy we calculated was 146 kilojoules per mole. Why do we care? Well, here's why we care. We can now use this activation energy, which does not change regardless of which temperature you're at. We can use this activation energy to calculate half-life. How long will it last? And here's what you get. The half-life of a collagen peptide bond at 100 degrees is 0.8 days. So half of them will break in less than a day. Okay, but it's going to get longer as the temperature gets lower. At 75 degrees, the half-life is 24 days. Half of them will be broken in 24 days. Half-life at 50 degrees is 3.2 years. Half-life at 25 degrees, which is probably about the temperature of that room you're sitting in right now. That's room temperature, 25 degrees Celsius, is 301 years. That's a long time. And this is good because this means the collagen in your body is not just breaking down with you just sitting there. It is very stable at room temperature. It's even more stable at lower temperatures, which I'll show you in just a moment. So here is the bone collagen decay rates in alligator. It has a half-life of 196 years at 25 degrees Celsius in a neutral aqueous environment. That's scientific speak. In the cow, it has a half-life of 301 years. In the ostrich, it has a half-life of 390 years. And think of these as approximate. These are estimates. That's what the squiggly line means. So don't focus so much on the number, but focus on the general number. So we're talking it lasts hundreds of years. So how long would it last during fossilization? Well, fossilization, where these fossils are found that have the collagen in it, it typically occurs between 25 degrees Celsius and 10 degrees Celsius. So the half-life at 10 degrees Celsius is just about 7,000 years. So let's use that because that's going to be kind of the upper limit for how long collagen could last in a smoldering in the grave. All right, so 7,000 years, what does that mean? Here's a graph showing the estimated life expectancy of collagen at 10 degrees Celsius. On the y-axis, I'm showing you the amount that would be remaining. On the x-axis, I'm showing you the time in years. So if you started with a fresh dead bone at time 0, 100% of the collagen would be present. Factoring in just thermal decay, which is what we're looking at. We're not factoring in bacteria. That would speed it up. immensely. We're not factoring in a lot of other things like pH that would speed it up. We're just factoring in thermal decay. This is unstoppable entropy that causes the breakdown. Okay, so based on thermal decay and a half-life of 7,000 years, after about 7,000 years, 50% would be remaining. Another 7,000 years, 25% would be remaining, and so on and so forth. After 7 half-lives, you get down to 0.781%. So you can come to this conclusion. It would take approximately 48,000 years at 10 degrees Celsius for the collagen to drop below 1%. This is a very interesting result, and here's why. Do you remember our question? How long can collagen lie a moldering in the grave? All right, so the answer to this question, how long can collagen lie a moldering in the grave? I can't give you an exact number, but I can tell you this much. It's not 66 million years. This is by conservative estimates. So not even close to 66 million years. This is a huge problem because you're digging up dinosaur bones, which are supposed to be 66 million years old, and you're finding collagen in them, which is not supposed to last more than 48,000 years. Wow. Okay, so what do you do about this? So scientists accept that there's collagen in these bones. So how do they explain it? Well, here's Mary Schweitzer. Here's what she says. See if you can fill in the blank. The persistence of still soft vessels, matrix, and cells and fossil remains millions of years old is unexpected. And if original, refutes, what do you think she says? It refutes models of degradation. Rather than saying, it refutes the age of the bones or the presumed age of these bones, maybe they're not 66 million years old, she says, well, I guess we don't know how things degrade. And so scientists have proposed many mechanisms to try to rescue this. Here's the most common one. It's called the cross-linking hypothesis. And the idea is that if you have iron in the mix when these bones die, so iron from the hemoglobin from the blood, then that's going to create these free radicals, hydroxyl radicals. That's what the HO stands for. And those attack the collagen and cause cross-linking to occur between the strands of the collagen, and you get increased resistance to enzymatic and microbial degradation. This would help to preserve it. That's the idea, anyways. So, is there evidence for this? Well, the evidence that this is based on is primarily from one study in 2014 in which Mary Schweitzer and in her lab, they studied some ostrich blood vessels, and they took these ostrich blood vessels and they soaked them in hemoglobin, that's the one on the left, and just plain old water. So iron in the one on the left and no iron in the one on the right. And what they found when they did this was that the ones that were exposed to hemoglobin lasted 2 years. That's this guy right here. The ones that were soaked in water lasted 3 days. And they conclude that this was a 240-fold increase in stability. Now, Mary Schweitzer is a good scientist, but this is not good science. And I'll tell you why. Because we know that collagen at room temperature should last for hundreds of years. And it decayed inside these blood vessels in 3 days. So really what they should be asking is, why did the collagen decay so quickly in the water? Not why did it last so long in the iron? They're asking the wrong question in this particular experiment. And I can tell you why. Probably it was because there was bacterial contamination in the water sample, because collagen should not decay in 3 days, but it did in theirs. So this was not a good experiment, but it is the primary experiment used to justify the iron hypothesis. Are you thinking what I'm thinking? I'm thinking we could test this experiment, this hypothesis ourselves. And so we did, using our the same setup that we used to test the decay of collagen. Here's what that looks like. We tested water. Here's what happens when the bone is exposed to just water. The line drops. That's to be expected. Here's what happens when the bone is exposed to a glutaraldehyde solution over time. The green line. The glutaraldehyde is an embalming fluid used to preserve dead bodies. And so it doesn't decay. Wow, this is pretty cool. Okay, so these are our controls. We've got something that doesn't decay, embalming fluid. We've got something that does decay, that's the water. So what about the irons? So the red line is the iron treatment, and what you'll see is that iron treatment does not enhance preservation. So that 240-fold increase, that's not an accurate representation of what iron's able to do. It does not enhance preservation. But we aren't the only ones who've looked at this. Here's another paper. They studied something similar. They took bones and they treated them with different concentrations of iron and other things. And here's what they found. Iron actually accelerated the decay in this test. So this is the bone that was exposed to iron. So iron, even though they will still say this is still the main thing that you're going to hear. Iron does not preserve collagen or bone integrity. So what do you think? Do you think they've tossed out the idea that these bones are 66 million years old? Well, here's another quote. I want you to try to guess what the end of this quote says. If these components are demonstrated chemically to be original, then a mechanism must exist to allow their persistence across geological time. When you see a word like must or had to, what you're seeing is a scientist who's operating under a worldview, a belief system, and not science. And they're welcome to do that, but just don't call it science, call it your belief system. They can't think of any other possibility. Let's go back to that question at the beginning. When did the age of dinosaurs end? The biblical timeline predicted that it ended 4,400 years ago, which would mean there would still be collagen left in bones. The evolutionary timeline predicted it ended about 65 million years ago, which would mean there wouldn't be collagen left in bones. So what can we conclude from this? The presence of dinosaur biomaterial is not consistent with the conventional assigned ages of these fossils. Period. We could also say the presence of dinosaur biomaterial is consistent with the biblically assigned ages of these fossils. So there you have it. I'm not trying to say that, whoa, we've completely overthrown the whole theory of evolution in one fell swoop. No, what I am trying to say is that when you compare these two models, asteroid, 66 million years ago, flood, 4,400 years ago, the science supports the Bible. And therein lies the conclusion. Now, I'm going to take your questions in just a moment, but I'm a geneticist by training. So perhaps you're wondering what's next? Well, here's what's next. We began to wonder if proteins are still present in these bones. What about DNA? So there's been tantalizing evidence published in 2020 that there's still protein inside of, or there's still DNA inside of dinosaur bones. So here's what you're looking at in this bottom row. These are emu cells. So they're from a bird. And you can see the red and the blue, those represent DNA. And then this top row are dinosaur cells. They're from a Hypacrosaurus. And you can still see a little bit of red and a little bit of blue, which they said in this paper, the identification of chemical markers of DNA in Hypacrosaurus suggest it may preserve much longer than originally proposed. So what's the next part? Well, in pursuit of dinosaur DNA. That's where we're going with this. And I welcome your questions about that. Here's why we do what we do. We're not trying to prove the Bible. If God wanted to prove the Bible, he could do that much, much better than we ever could. But we are just trying to glorify God through science. Thou art worthy, O Lord, to receive glory and honor and power, for thou hast created all things, and for thy pleasure they are and were created. These are the folks that made this work possible, among others. There's others not pictured here. This is the research team at the Van Andel Creation Research Center. Kevin Anderson was the former director who died unexpectedly and started this project, and I picked up his work from there. Brian Thomas was a huge supporter from Institute for Creation Research, and then our CRS supporters. So all of this research is made possible by donations from God's people. We don't receive government funds, obviously, to do creation research. So thank you. And with that, let's take some questions.
So we've grown up all of our lives being told that petroleum comes out of the ground from dinosaurs. Where does petroleum really come from?
So petroleum is a hydrocarbon, and so it comes from organic life forms. If a dinosaur got squeezed and decayed under enough heat and pressure, it could form into oil or petroleum. But the primary source for that would be plants, and lots of plants. And so, where it came from from the biblical perspective was the pre-flood world would have been just it would have looked like a jungle compared to our current world, which is filled with deserts and Antarctica and frozen stuff. The pre-flood world would have looked like a jungle. So it would have been very rich with plants. And then the flood comes along. The flood comes along, rips up all those plants, buries them under a lot of pressure. A lot of pressure that creates a lot of heat. And that's all that's required for the oxygen to be removed from these plant materials, and that turns it into coal and petroleum hydrocarbons. So that would be a product of the flood.
Okay, thank you.
Yeah, good question.
Hello? Can you hear me? So, I know we looked at water, it can increase degradation, fast. So what happened during the flood? Weren't the bones in the flood for you know, for a long time? Why didn't they degrade quickly?
So the reason we looked at water specifically is because I'm going to pull up my little model here. It's not actually because water does anything special to collagen, but water will do this to any biological molecule. Those little bonds right there, all that's needed for that bond to break is water. Water will get in there and it will split these apart. And this is why uh, over time, entropy, everything breaks down because all you need is a little bit of energy and a little bit of water and it will all break down. So it's not that water accelerates the decay. Um, Water is the molecule that breaks the bond. And so in the flood, of course, when these bones would have been laid down, they would have been laid down in water-borne sediment. So they would have been loaded with water. So there would have been plenty of water for this decay to occur. Furthermore, in the evolutionary worldview, so let me speak as an evolutionist for a moment. Um, they still require water for fossilization to occur. So these bones would have been laid down by water in their worldview as well, just not a global flood. And then that water, the groundwater, flows through these bones over hundreds and hundreds of years and gradually replaces the bone mineral with rock mineral. So the bones that you dig out of the ground are actually no longer bone. They're rock that is in the shape of the original bone. That's what fossilization is. So I said a lot there, and I say all that to say this. Um, when these bones are buried, regardless of which worldview you have, they're buried with a lot of water present, and water is all that's needed for collagen to decay.
You talked a lot about collagen. What other biological molecules have you found? Lipids or sugars or anything like that? I think Armitage had said that he found some other molecules. Can you speak to that?
So we have not we have not looked for those. As a molecular biologist, my focus has primarily been in DNA, which is one of the major macromolecules, and protein. But the two that you mentioned, lipids and sugars, we have not looked at those. Our techniques would not detect those, the techniques that we use. Other researchers, though, have found chitin, and which is a carbohydrate. Um, they have found sugars, carbs, and not so much lipids. That's not one that's found a lot in these ancient fossils.
How fast does the fossilization take? Have they done studies on that? And where would I find the information on it?
Um, so there's an interesting fella recently. His name is Gordon Craig. He's done fossilization, accelerated fossilization using a lot of heat and a lot of pressure. So the name's Gordon Craig. And it's really neat what he's done. He's produced these fossils and then he's posted pictures of them. And paleontologists will look at them and say, "Oh yeah, that's a fossil from this rock layer. It's, this X millions of years old." And he just made it in his garage. Um, So it's not so much about the time, and even the evolutionists recognize this now. They everything's about time for them, but it's not so much about the time; it's about the conditions. The conditions are far more important than time. And so heat and pressure is one thing that will quickly make fossils and the coal and petroleum that was talked about earlier. The other thing is the amount of water flowing through an area. So it's the water that's moving the groundwater that's what's replacing the minerals. So if that fossil has a lot of groundwater, then it would be replaced pretty quickly. If only there was like a mechanism in the biblical worldview to produce a lot of water that covered the whole earth at one time.
Thank you for your talk. How much collagen do you find in dinosaur bones, and does it change depending on the region and temperature?
Oh, okay, that's a really really good question. So it absolutely, there's not a single comprehensive study that has looked at this, but you do find it's not in every bone. So some bones, especially those in warm, temperate climates, the collagen doesn't last as long. And so those bones are often devoid of collagen. Now, what we're very interested in is colder bones. There are bones in Alaska that actually have not been fully mineralized, dinosaur bones, which have not been fully mineralized because it's frozen up there most of the time in these particular areas. So the bone is still fresh, so to speak. And those bones we would predict would have lots of collagen. We've never got our hands on those, but we're working on that.
The program that you showed today is pretty well proved a creation. And my question is, what kind of leg do the opponents have to stand on for that? The evolutionary people, they can see what you're coming up with, and what is their take on that, and how do they say that you're wrong?
Okay, so well, for starters, this is unpublished data, so we're working on publishing it as we speak, and we're not trying to publish it, and let's put it this way, we're gonna let people draw their own conclusions from it. But I'll tell you ahead of time what the reaction will be. It will be, well, maybe iron doesn't preserve collagen, but something somewhere out there does. And let me tell you a story or an analogy to help illustrate the response. There was once a man who went to the doctor and he said, "Doctor, I'm dead." And the doctor said, "No, you're not. You're standing in front of me." And he said, "No, no, doctor, I'm dead. Can you perform a test on me to prove that I'm alive?" And the doctor said, "Yeah, yeah. Dead men don't bleed. So if I cut you and there's blood, then that means you're not dead." And the man agreed. He said, "Okay, let's see." So the doctor pricked his finger, and sure enough, blood came out. And the man said, "Well, I'll be. Dead men do bleed." So you see what's happening there. So that's exactly what's happening in this field. You look at all the evidence, and the evidence says proteins don't last that long. And then they dig up a dinosaur bone that has proteins in it, and they say, well, I'll be. Proteins do last that long, in spite of the evidence.
You can get some steps in. Another question: how many different orders of animals has collagen been found in, and how deep in the geologic record? What's the lowest or oldest, as evolutionists would say?
Okay, so I don't know the answer to that. It is a vertebrate specific protein, so it's found in all vertebrates. Um, but it would not be found in invertebrates like the trilobites and things like that. It's been found down to, on the conventional timeline, down to 180 million years. They can find remnants of collagen. Now that is, of course, using their timeline. From our perspective, it should be present across the geologic column wherever you find vertebrates, but that depends on the conditions in which those vertebrates are buried. So things buried lower would be under more heat and pressure, so there would be less collagen in those.
I've got a question for young people that might be watching this in the future. If they wanted to do what you're doing or assist you in what you're doing, what would you suggest they do from the time that they leave high school and into college?
Okay, so I believe that God has gifted certain people with that knack of curiosity and the desire to figure out how things work. In science, that's a gift from God. And unfortunately, science has been overrun by atheism and materialism, and that's a shame because science is not the bad guy. It's those worldviews that are brought into it that are the bad guy. And so I encourage young people to pursue science. And here's what that looks like. If that's you, if you love research, if you love studying how things work, um, after you graduate high school, I recommend attending a Christian undergrad university. You'll have a lot more support there. But then while you're there, you need to start doing some research during your summers. Do research internships at regular secular universities. And the vast majority of science is just good science. So just because it's done at a secular university does not mean that's all bad. So I went to the University of West Florida. We studied Alzheimer's disease. Fantastic. Went to Cornell University where I studied development. All that is just pure good science, and you can get that at a secular university. So pursue that training all the way to that terminal degree. You can get just some incredible opportunities by pursuing the PhD level research, and then see how God can use you. Along the way, make sure you stay connected to a strong church. And I've had my wife with me through this all, and she's been a really great support and anchor for me.
Great, thank you. Say, we have one question from online that was written in the chat. Have you carbon dated the material?
We have not, but other creation researchers have, because these are carbon-based molecules. So there's carbon in them, so you can carbon date them. And from a half-life, actually the half-life of carbon is very similar to the half-life of the peptide bonds. It's about 5,700 years. And so it also should be gone well before 100,000 years. And you find carbon 14 in these bones, and you also find carbon 14 in other things such as coal and diamonds, which are also ancient in the conventional timeline and should not have carbon 14. So it corroborates and matches these collagen results. And you can look at the Creation Research Society quarterly. Some of that research has been published in the Creation Research Society quarterly. Most of that's freely available online. Go to creationresearch.org. As we're wrapping up, I'm going to put up that the, the screen there. On the left-hand side, that's for everybody. If you just want to kind of know what's happening in creation research, uh, scan and join our newsletter. Membership this is a paid membership. This is one of the ways the functioning of the society works. So if you are a scientist, we definitely want to recruit all scientists that are Bible believing Christians to be part of this society so we can work together. But even if you just love the technical side of creation research, you can be a sustaining member of the Creation Research Society. Thank you so much for your attention and for your great questions. And I look forward to seeing you all in person perhaps sometime.